So I'm going to tell you about a tool that we developed recently that allows us to work with non-model systems. And specifically in this case, what we are interested in doing is tagging genes in their native context, their native genomic context. And we wanted to use CRISPR-Cas9 and repair templates to do this. But the challenge with working with these non-model organisms, for example, cassava, one of the plants that I work on, is that there aren't a lot of tools available. If you want it, you have to make it. With cassava, we're lucky that we do have a fairly robust transformation pipeline. But as you can see here from the time scale, this is really a long-term commitment if you're going to make something. In addition, we have this challenge that in plants, as I'm sure many of you know, homologous recombination is a very rare event. And many of the genes that are really interesting to us are expressed either at very low levels or only in specific tissues. And so this makes this whole goal very challenging. A little bit of background for the gene that we wanted to tag, it's called Me-sweet10A. It's a sugar transporter that is normally expressed only in flowers and cassava. However, it's also topically expressed by a pathogen that we study during infection. So here, this bacterial pathogen is injecting its own proteins into the plant cell, some of which act as transcriptional activators or tau effectors to activate host susceptibility genes. And while we've known this happens for a long time, the spatial and temporal dynamics of this process are not clear. So we don't know whether or not this gene is activated in lots of different tissue types or exactly when during the infection process it's activated. So we figured if we could tag Me-sweet10A in its native genomic context with GFP, that would be a useful tool for uncovering the answers to some of those questions. And I appreciated Tessa's comments earlier of how nature often gives us the tools that we need to do what we want. And that was the case here. So Ahuma Okwano and Kira Vili, two people in my lab, realized that they could use that transcriptional activator from the pathogen to activate expression in tissue culture of the cells that they wanted. And so what we did using transcriptomics data that had been generated in my lab a while ago, Kira identified a promoter that would drive expression only in the tissue that we use for transformation, friable embryogenic calli, and then constructed a very large tDNA that included our CRISPR-Cas9 reagents, our repair template that spanned our region of interest, that included GFP, and then also this FEC specific promoter hooked up to Tau20, which was our effector from the pathogen. These reagents were introduced into cassava FEC cells, and then during the tissue culture pipeline, very early in that process, we were able to screen for GFP positive cells and isolate specifically those cells to regenerate cassava. And so really the important thing here is that it allowed us to identify super rare events. The really exciting thing came when we actually were able to regenerate cassava plants from those few initial cells, and then using RT-PCR and confocal microscopy demonstrate that we had in fact functionally tagged Me-sweet10A in its genomic context, and that after inoculation with the pathogen, we were able to see expression in planta. Now, throughout the course of this project, I ended up talking with my colleague at the Danforth Center, Blake Myers, a fair amount. And for those of you who know Blake, you'll know he's quite the ideas man. And we just really had a great time talking about all of the different ways that we might be able to use programmable transcription factors, for example, designer tau effectors or deactivated Cas9 fused to transcriptional activators combined with CRISPR to use this strategy to target any gene. And so we decided to call this Surefire because you're surely going to get what you're looking for. And so some of the things that we've been talking about is, well, using these C-terminal tags, we can ensure functional expression at that early stage. That's certainly a time-saving advantage. So what would you do with a genomic tag of your favorite gene? Using N-terminal tags, it should be possible to replace promoters. So what would you do with an inducible native context version of your favorite gene? And also we think that using this technology, it should be possible to tag multiple genes simultaneously. So what would you do with a tagged complex? And we were instructed to try to leave the group today with questions. So I'll just leave you with what would you do? Now, just quickly, I'd like to thank Kira Veely, who's a senior research scientist in my lab and really has been leading this project, as well as Ahuma, who is located in Nigeria, but got to spend about 8 months working with us. They were really the ones who came up with this initial work. And then more recently, working with Blake Myers and his team, funding through NSF EDGE program to really customize these tools and generalize them. Thank you.